In the first step of meiosis, called interphase, DNA is replicated and it doubles the available DNA for cell division. In the second step, known as prophase 1, DNA condenses into chromosomes. The nuclear envelope also begins to dissolve. During this stage, chromosomes can cross over, creating variants in DNA sequences. A chromosome is 2 chromatids. At the end of prophase 1, a tetrad is formed with the 4 chromatids. Kinetochores connect to spindle fibers to help guide chromosome movement. At metaphase 1, the crossed over tetrads line up in the center of the cell known as the metaphase or M plate. Just like a knife cutting apart two things, in anaphase 1, our spindle fibers will pull apart these 2 chromosomes and lead to our next phase and put these 2 chromosomes on opposite sides of the cell. Up here, pulling apart attached to the spindle fibers are centrioles, which consist of 2 centrosomes. Next, we have telophase. At this point, nuclear membranes begin to reform and the cell begins to divide. This line between the cells is called the cleavage furrow. This is the beginning of cytokinesis. And like the 2 cells are pulled apart and they pop together and they pop apart, we have cytokinesis, and now we have 2 haploid cells. If you notice, we will have 2 rounds of division in meiosis, while in mitosis there's only 1 round of division and only 2 cells are produced. So next we have interphase 2, which is actually known as interkinesis because no DNA is going to be replicated. And this will result in the production of haploid cells in the end. So each of these are 1 chromosome, and that chromosome consists of 2 sister chromatids. This phase, we wait for time, just like it happened before interphase 1. Now, during interkinesis, we are just waiting for time for DNA to just sit there and pass through its different checkpoints. Moving on to prophase 2, the centrioles move to the sides of the cell again, nuclear membranes start to dissolve, and the cell is overall preparing for metaphase 2. Your DNA condenses again into your chromosomes and starts to move around. Next up is metaphase 2. The same thing that happened in metaphase 1 happens in metaphase 2. Your singular chromosome will line up on your metaphase plate, and then the spindle fibers will attach, ready to pull apart. Moving on to anaphase 2, the spindle fibers pull on those chromosomes again, pulling apart the 2 sister chromatids. Now we have 4 different areas, each with their own DNA. Moving into the final phase, telophase 2, we have 2 new nuclear membranes form in each cell, and then of course cytokinesis, separating each of our 2 cells into 4 different haploid cells. And this right here, this is our result of meiosis. So we have 1 cell produces 2 cells, which each produce 2 cells, meaning during meiosis, 1 cell will become 4 haploid cells. And that is meiosis. Thank you.